So the next part will be the talk from Dr. Nathan. Please share your screen, please.
Yes, give me a moment. Can you see my screen?
Yes, please make it full screen, please.
Sure. All right.
Yeah, I can see your full screen.
Okay. All right. So not to further ado, let me start. So for the one who don't know me, my name is Thanet Jantadet. I'm based in Thailand. My position is the Regional Application Support Specialist. So the topics today, we will go through the comprehensive solution for the oncology NGS, including the Chi-seq targeted DNA, RNA, and the multimodal DNA and RNA. And actually, we just launched the long lead kit. So we will add it for you in the last part as well. So let's see the agenda today. So we separate into the two parts. So for the part one is the overview. We will go through the cancer incident in Vietnam, molecular genetic testing for the current, and sequencer platform and the Chi-seq compatibility. Then we will go to the second part later. Let's go to the first one. So as you can see, I got this number from the WHO 2022. It's quite a bit old, but that is the update one. I can search it for now. Anyway, as you can see that the number of the new case now is almost up to the 200,000. Number of the pass away people that caused by the cancer is around 100,000 as well. And if we count 5 years, it's almost 500,000 that related to the cancer affect to the life for the life quality for the Vietnamese people. So, but anyway, if we think in the positive way, this number maybe it's happened for 5 to 10 years already, but now Vietnam have developed and upgrade about the instrument and technique to screening the cancer more and more advanced. So that's why you can detect more and more number of the cancer incident and the number of the people that pass away by the cancer. So anyway, this cancer, we believe that finally, since we can detect the early stage of the cancer, so now with the power of the precision medicine, we expect that we can treat the cancer people with the precision medicine as well. And QIAGEN have that solution. We will go through it today. So if we separate by gender, for the male in Vietnam, the number one cancer is the liver, lung, and the stomach. For the female, is the breast, lung, and the colorectal. For the both sex, the number one is the breast cancer and the liver, which come from the man. Yeah. So now, I try to find another information and found that the risk key factor actually is come from the first of it is come from the infectious agent. HBV is the cause of the liver cancer. HPV is the cause of the cervical cancer. Tobacco, definitely lung, throat, and another cancer that related to the respiratory tract. Alcoholic consumable consumption related to the liver, colorectal, and another digestive tract. Yeah, and environment as well. PM 2.5, definitely a lot now around Southeast Asia is the incident for the lung cancer. By the way, I know that Vietnam government try to set up a lot of campaigns to reduce this kind of the key risk factor. For example, for the infectious agent, you have the vaccinate program to reduce the HBV case that related to the liver cancer. You also have the tobacco control. And importantly, you also collaborate with the international partner, which the WHO, that they concern and set up about the national cancer control strategy via genetic testing. So the take-home message here is, if we know what drives the tumor at the genetics level, how might that change the way we diagnose, treat, or even prevent the cancer? So when we talk about the genetics level, the current protocol is the PCR. Some lab use the microarray as a gold standard. Some lab use the Sanger, or even some lab now use the NGS or the next generation sequencing already. each method have the pros and cons. I will not go through it in detail, but just focus only the NGS because that is the technology that we will go through it today. Yeah. Next generation sequencing, even though it's the high throughput, when we talk about the high throughput, meaning that you can do the sequencing, you can get the sequencing data for the multiple sample in at the same time of the running. However, to get that one is not easy because it's very expensive technology. You need a very high budget. It takes turnaround time quite a lot. Maybe from end-to-end process is around 5 days. Comparing with the different method, maybe you can get in 1 day. Yeah. However, you also need the skillful technician as well that know how to do the library preparation and the bioinformaticians to analyze the challenge data that come from the sequencer. However, that solution, that issue, we have the solution from QIAGEN today already that make your life a bit easy for the library preparation in the wet lab part. Also, for the software during the secondary and tertiary analysis part, which will be presented by my colleague in the next presentation, Dr. Shiva. Yeah. So the generation of the sequencing, if we separate now, we can separate into the three generation. First generation is the Sanger sequencing. So some of you may know this machine already. Yeah. Second generation is still can use until now, which is the short read. And the third generation is emerged in the market more and more this year until last year already actually. So the second year and the second generation and the third generation, this is the up to date uh, sequencing technology that's still available, and QIAGEN have the solution for both second generation and the third generation already in terms of the wet lab and analysis part. So if we go to the um, sequencer platform. So as I told you that we have the two separate type of the sequencer. On your left is the long read one. As you can see that PacBio have and the Oxford Nanopore is the main company that related to the long read. Meanwhile, for the short read that you may familiar with the sequencer platform is the Illumina MGI S5 from the Thermo Fisher, GenMine. PacBio also established the short read as well recently, which is the ONSO, yeah. And you may heard about the Element and the Singular and the Ultima, which three of these one used in the Western and the EMEA country. So now let's see for the sample to insight or the workflow that QIAGEN already have the solution. So the first presenter also tell you about the sample preparation already. That is the first part that you have to do by extraction from the raw sample, even though it's the fresh sample, blood, serum, or even the cell-free DNA, urine, saliva, we have the solution already. Even you want to do the by the manual or by the automate, we have the solution already. So I will not go through it on this. Once you have the sample preparation, we need to do the sample QC. So the sample QC, normally what you need to do is check the concentration by the qubit or yeah, I recommend use the fluorescent method, and you check the purity by the QIAexpert, and then you do the library preparation, depend on the panel that you interest, and once you finish the library preparation, you have to do the library QC. Normally, what we need to check is the concentration and the size. So QIAGEN have the QIAxcel to allow you to check the library size and the primer dimer contamination as well. All right. And then we have to go to the primary analysis by the sequencer, which this is only step that QIAGEN don't have. QIAGEN normally we ever have the sequencer, but it's obsolete already. That one is the gene reader. So that's why we try to develop the library preparation kit to make it compatible to all sequencer type which available in the market, even though it's the short read or the long read. we can run in all the platform now. Yeah. So that's why for the NGS kit, QIAGEN use the U in front of the UNGS kit, which come from the universal, meaning that our library prep kit can uh, run in compatible with the all type of the sequencing platform by now. Yeah. Once you finish the library prepare library sequencing, you will get the raw file FASTQ. Go through the secondary analysis by the software that QIAGEN have CLC analysis. You got the variant calling profile in the VCF format. Then you go to the last part, tertiary analysis, for annotate the data until get the clinical report. So the doctor will know which precision medicine that available in Vietnam and should treat to the particular cancer patient. So this is overall from end to end that QIAGEN already have the solution. Yeah. So just want to give you a brief idea on the first paragraph here. This is the name of the main QIAseq product. When you heard about the QIAseq, it means the name of the library preparation product. Yeah. And on your right here is the name of the sequencer. The green one meaning that is compatible and we already validate on that machine. Yeah. For the yellow one is the one that compatible, but we not yet validate. Meanwhile, the red one is the one that not compatible. So as you can see, the red one is related to the long read sequencer, which most of the kit is not available, but just only one, which we just launched this month, I would say, which is the Chi-seq X-type long read panel. We will go through it in the last part, yeah. And another platform that got a lot of the red light is the Ion Torrent, which is the single read. So the reason that it's not compatible because of the most of the panel we have is quite huge. So that's why the capacity of the chips for the Ion Torrent maybe it's not enough, but for some application, for example, especially for the onco solution here, fusion for the RNA fusion detection and the microbial is available for us to detect. Okay. So
It's the activity.
that's it. Finished for the first part already. Now we go to the second part, which related to the end-to-end workflow analysis guideline. And the new panel that we have is the CGP panel in the long read solution. All right. So let's go to this page again about the sample preparation. Just want you to get some idea that for the extraction part, we have the solution for the DNA-seq application, cell-free DNA-seq application, RNA-seq application, or even the multimodal, which allow you to prepare the DNA and RNA library simultaneously. So all sample type for each application we have available here already, even though it's the standard DNA from blood, FFPE, or even the cell-free DNA. We have all for the each type of the application already. Okay, so if you want to get more detail about the each extraction kit, you can text to me later or you can contact to the marketing team or even the first presenter who is the expert for the extraction kit. So now we go to the library preparation part. So I designed to separate by the cancer analysis guideline. Normally in Thailand, we separate into the three part, which is the low risk of the individual person, intermediate risk, and the high risk of the cancer patient, I would say. So let's start with the low risk. Low risk is the individual with the family which has the cancer. So when we talk about the history of the family who got the cancer, what what they want to detect is related to the germline, yeah, not the somatic, but the germline itself. So um, the risk reduced. So it depends on whether after they detect by the next generation sequencing, they can see something or not, or some signal or not. So we have the QIAGEN Comprehensive Hereditary Cancer Research Panel, which combine several types of the cancer and have the gene list around more than 300 gene. Yeah. So it depends whether they can we can detect any cancer or not. If not, we still able to explain to the individual person that you can come to the hospital and get the body checkup every year based on the age, based on the gender and the sex, which I will show you later for the criteria. Yeah. So before that, let's go to the gene list that I just explained you for the low risk individual person. So the name of the kit is here, Hereditary Cancer Research Panel. Is composed of the 317 gene, can detect 12 cancer, and these main 12 cancer also have the 124 cancer subtype. Yeah. If you want to know more about what type of the cancer and which gene related to what cancer, you can go to this link. Okay. So where exactly is located on the our panel list? So is here comprehensive hereditary cancer research panel that I just suggest you for the screening the low risk purpose. You can run in the MySeq using the correct depth around 500x. You use the V3 flow cell type, you can run up to 7 sample. Yeah. But we also have the several type of the panel which allow you to detect the germline. Yeah. So later on, you can come to this slide and check whether it's catch your interest or not. The good thing is that let's say that you interest about the colorectal cancer panel for the germline detection, and you want to modify it from this catalog panel by adding or delete some gene. You can do that. We have the customized panel. So all of the kit here is the customizable. All right. So this is just some guideline in case that the customer join or apply to the low risk program that's set up by the hospital and they cannot detect anything. So you still able to suggest them to do the body check separate by the gender and the age, range of age. So that is the way that the hospital still can follow up with the customer. So in terms of the intermediate risk, so normally we suggest with the one that probably have the healthy, didn't see any signal or they feel healthy, but once detect the blood, they can detect some things to indicate that hey, you have a low risk, you have the some risk of the particular cancer. So this one is related to the somatic. It's not the germline anymore. Yeah. So we have the solution named QIAGEN, Human Actionable Solid Tumor. When you heard about the actionable, meaning that the biomarker or the gene list in this panel is we is known is the one that we know available clinical action, meaning that we know the precision medicine list for this kind of the uh, gene list in this actionable panel already. Okay, so that's why for the individual people that join this program based on the indicator from the blood, they can go up to the precision medicine. Yeah. So if you're curious that what indicator I mentioned, so here I list for you already. This is the list of the indicator. I will not go through it in detail, but from time to time and many publication now they know that several indicator is related to the specific cancer type as well. So we detect from this for the intermediation program. Yeah. So now when we talk about the gene list for the actionable solid tumors, this panel composed of the 30 gene list, which composed of the actionable gene list related to the several cancer as listed here. Anyway, as you can see that for the colorectal, there are just only the 5 genes. And for the another type of the cancer, they also have the different type of the gene, but it's not that much because the purpose is just only for the screening. Yeah, whether you have based on the indicator that we can detect whether you have the any risk of the actionable gene that appear in this panel or not. All right. So where this kit locate is located in this page. Okay, it's the different chemistry we call the V3. Yeah. So as you can see that apart from the actionable solid tumors, we also have the different type of the panel based on this chemistry, including the cancer related and the non-cancer related. The non-cancer related that I mentioned is the, for example, the mitochondrial panel. We also have the pharmacogenomics. We also have. Yeah. And this one, I guide you the sample per run in the MySeq, NextSeq for the somatic that fix the variant of frequency at the 5% and the depth at the 7,200. You can run up to this number. Yeah. Based on the different type of the sequencer and the flow cell use. Okay. Anyway, if your lab have the different type of the sequencer, could be the MGI or Element or the PacBio, also, and you want me to calculate for the sample multiplexing, I'm happy to help. Just text me, yeah, or send me the message later. So now we go to the high risk. Who gonna be join the high risk is the one that individual that already diagnosed with the cancer. They know what cancer they got already. So definitely it's go to the somatic. And we have so many specific onco cancer panel. For example, if they know that they got the lung cancer, so we have specific lung cancer. This one will be better than the actionable because actionable, we also have the lung cancer, but it may have the actionable gene just only the 5 to 10 genes, not more than that. But if you have the specific onco panel here, you have the gene, I think more than 100 genes that related to the lung cancer, which allow you to get the um, greater number of the precision medicine choice, whether so it will be more specific and more choice for the doctor to consider whether which one should treat to the patient. Okay, so this is some just brief idea for the low risk, intermediate risk, and high risk, which kit should fit which application. Okay, and where it sit into the portfolio. So this is under the different chemistry is V4. With the power of the V4 chemistry, you can finish the library preparation in half day. Because if you know how to do the library preparation, the tedious part is the cleanup step. So we not use the cleanup step with the bead cleanup anymore. With the power of V4, we use the enzymatic cleanup. So we engineer the enzyme to recognize only the short fragment, which normally is generated from the artifact and the primer dimer. So that engineer enzyme will digest that one. And the output, you can go to the next step. So that's why the turnaround time is very, very short. Once the customer change through the V3 to V4, they don't want to go back to the V3 anymore because the turnaround time for the V4 is pretty much shorter comparing with the V3. And you can prep and start run the sequencing in the same day. Yeah. So what panel we have? The one that I just showed you in the previous slide is here, somatic hotspot. Yeah, breast cancer, colorectal, myeloid, brain, and the lung. However, all of these cancer, we also have for the somatic full coding as well. But the full different thing for the full coding and the hotspot is that the full coding will cover for the whole exon. Hotspot will cover only the particular nucleotide that we know already that is the uh, um, uh, uh, tend to be the pathogenic mutation if there is some change on that hotspot. Yeah. So it depends. If you want to detect the hotspot, not the full coding, definitely you can run more sample per run in the same flow cell. For example, breast cancer, for the somatic hotspot, you can run in MySeq V3 up to 11. But the same panel with the full coding, you can run just only 3 because if you want the full coding, you need more read per sample in the equal flow cell. Yeah, comparing with the hotspot. So this is some idea that I just want to give you for the targeted DNA. Anyway, if you think about something bigger, something huge, more than 500 gene list can detect anything for both DNA biomarker and RNA biomarker. We now have the solution. The name of the kit is the QIAGEN multimodal. DNA/RNA library prep kit. So this is the backbone chemistry, and they enrich by the probe hybridization. And the probe that we have now, the huge one called the CGP panel. CGP comes from the comprehensive genomic profiling. How the workflow look like? All right, please allow me to skip this one because I afraid that it will spend too much time for you. So what is the CGP composed of? So it can detect the DNA biomarker up to the 724 oncogenes and tumor suppressor gene as well, meaning that the TMB and the MSI we also can cover. Copy number variation and loss of the heterogeneity we also can detect. Yeah. DNA translocation, we also can detect. So that is the DNA part for all that we can detect. I believe that this kit is the biggest one for the current library prep kit that available in the market. Yeah. We also can detect the RNA marker. Normally, when we talk about the RNA marker, it will including the expression and the fusion. Fusion that we can detect is including the known and novel fusion as well. So we have another 234 genes. Some genes here in the RNA is overlap with the DNA, but not all of them is overlap. Yeah. If you interest about the gene list for the CGP panel, what it composed of, you can send a message to me. I'm happy to share. Yeah. Cancer type that we can detect is available in this list already. Okay. So now let's see the workflow, how the multimodal allow you to detect both DNA and RNA. Yeah. Start with input material, you can add the total nucleic acid, which composed of the DNA and RNA, which the last presenter also mentioned about the all prep kit, which allow you to extract the DNA and RNA from the same sample. But that DNA and RNA will available in the separate tube. Okay, so it allow you to detect the concentration and do the QC for the DNA part at the end the RNA part as well. Yeah. So that is the good thing of the all prep kit. Yeah. So let's go very brief for the workflow. In the beginning, you have to insert the DNA and RNA here, okay, which you can do the concentration check and the QC check as well, as I told you before. So I count it as a total nucleic acid, yeah. So we start with the fragmentation for the DNA and the fragmentation for the RNA by the heating. DNA will do the end polishing by the do it blind end and A adding, and then all RNA that fragmented, we will do the A adding, poly A adding, yeah. And for the DNA now, we do the adapter ligation. And from this step, you will have the complete DNA library already and the RNA that not convert to the cDNA yet. So we split this one into two parts for the DNA part and for the RNA part, okay? For the DNA part, is ready to go to the whole genome sequencing. If you want to get the whole genome sequencing data, you can use the library prep from this step to run sequencing directly, all right? But for the RNA here, now there is the DNA and the RNA, right? So we will treat with the DNase to eliminate the DNA library and remaining only the RNA, and then do the reverse transcription to change only the RNA to be cDNA. And then that cDNA will do the indexing and is ready to go to the whole transcriptome sequencing. If you're interested to generate the data for the whole transcriptome sequencing, you can go to the next generation sequencing. Anyway, if you want to narrow down, interesting, just only the gene list in the CGP panel that I just told you, we can do the hybrid capture at this step. That probe, we can do the customization as well. And also is compatible with the third-party probe for both DNA and RNA. DNA, the third party that I mentioned is available here from IDT, Kappa, Agilent, and Twist Biosign. You can use with them to capture the target of interest by this step for the DNA and at this step for the RNA-seq as well. Or you want to combine the CGP panel with the third-party panel in the same time to capture for both target gene in the CGP panel and the third-party panel also can do it. All right. So if you're curious that you need to if you interest only the DNA part, but you don't interest about the RNA part, you need to purchase two of them or not? The answer is no. You have a choice. You can purchase only the DNA, the backbone chemistry, just only the DNA, or backbone chemistry, only the RNA. As shown here, this is the QIAGEN page. As you can see here, there is the CGP DNA, CGP RNA available for you. So it's not force you that you need to purchase the DNA and RNA. All right. So it's quite very, very flexible here. So by compare with the competitor, we can detect more. Now we are the best one that can detect more list of the DNA gene and the RNA gene at the same time. Customizable is available. You can finish the library prep in 1 or 2 days. It's available for the workflow for the FFPE and the cell-free DNA. Input DNA is the 40 nanogram. And the best one is that we guarantee that the price per sample is cheaper than the different company for sure. All right. So now is the long read that we just launched last week. So, all right, I will speed up. Um, we have the two panel for the long read now, which compatible with the PacBio long read platform and also the nanopore. So if you have this name of the sequencer platform available in your lab, you can run these two kits, which is the hereditary cancer research panel and the HLA typing. Let's go to the, okay, I will skip this one. Let's go to the hereditary cancer panel first. So this is the list of the gene for the hereditary cancer for the long read. We have up to the 95 gene which related to the structural variant. That's one you can detect, and we do validate in PacBio already. It requires just only the 0.4 million, and you get the 40x coverage. If you go up to the 1 million per sample, you will have the 100x coverage. What type of the structural variant you can detect? Long insertion, long deletion, inversion, interspersed duplication, translocation, and the tandem duplicate. All of them we can detect by using this long read kit. All right. Now we go to the HLA. HLA, we cover for both HLA class 1 and HLA class 2 is cover up to the 18 full length of the HLA gene, which cover not only the coding part, but also the intron and the UTR as well. So for the one that doing the application about the HLA, I believe that you will be happy on this. Okay, and the application that use is the transplantation, immunogenetics, and the disease associated study. All right. So this is my last slide. So the kit that we just go through it today is just some part of the targeted DNA, yeah, and the targeted RNA is fusion here, but and also the multimodal here, okay. But apart from this, we also have much more kit which related to the microbiome. For example, if you want to do the whole genome sequencing or the bacteria virus, we also have the solution. You want to study the 16S expression profiling for the microbiome, we also have the solution. Epigenetics for the methylation and ChIP-seq, we also have the solution as well. So if you need more information or any additional data of the sequencing, do not hesitate to contact me or my colleague, Mr. Tang. I provide you the mail here. All question is welcome here. Thank you.